and for some of you, and let's have Veronica's third lecture.
Yeah, third lecture. Okay, so great that everybody could make it on the national holiday. So this morning I wanted to start a little bit more easy with some more abstract ideas about pattern formation. I think this is the picture, which is, Ricardo, I think you guys use this as a background, something like this, right? So I actually woke up this morning and thought, let's talk about these patterns, okay? So I was telling you a lot about the roots, and if there's a little bit of time, we can go back to that. But those patterns are very influenced by the local stimulus that they are noticing. So there's a lot of decisions to make there. So very differently than this humanized plant, which has a very clear Bauplan like ours. Plants actually, every time you would grow the same genotype, the same clone, you would get very different root architectures because you can't control for all the variation. However, what's happening above ground is more stereotypical, right? It's more repetitive. And that's something which has fascinated mathematicians. So when we go have coffee upstairs, did you guys notice that there are lots of plants there? The lady who makes the cake, I was talking to her, she likes to take little leaves and she propagates them. And she has this variety of plants there. And if you go talk to her, she really knows a lot about plants because she looks at them. And this is something that I just want to tell you guys should also do, like she does. You have to look at the things around you and observe, because there's still quite a lot of questions to ask. This here, for example, I don't know exactly how that pattern is, because it's not really the veins, it's something else, but there we go. So, someone who also looked a lot at these patterns was Astrid Lindenmayer. He was a Hungarian botanist who came to Utrecht, and he began the group of theoretical biology. So he is actually my PhD grandfather, because his student was Pauline, and Pauline was my supervisor. So there was an asbestos problem in the university, and we had to clear up some of the closets. And I came on to his writings and many, many sheets of computational simulations and pictures. And in those days, the pictures were not as we had them today. The computer graphics was very different. Punch cards and all sorts of stuff. So one thing is that if you start looking at those patterns that we see with our eye, we should realize that these sheaths of leaves, they're being formed, right, at a primordial stage and already having some type of alternating patterns. And the way that this is laid down will afterwards, unfurl in those macroscopic positioning of the leaves. So what am I saying here? Leaves will come, you will see, they can be alternating, they can come in certain angles, they can go 2 by 2s, or they can go in whorls, okay? Although you might think, okay, this is something specific for plants, if we go to other animals or other systems, like even the dictyostelium, which we will see tomorrow, we're going to see the simple one, but there's something called the polyspondylum, which has whorls also. So these type of patterns are, they will emerge in many different systems, yeah? So, this phenomena of leaves arranging in these certain mathematical patterns we call phyllotaxis. And you can also get these beautiful things like that you tend to see in cacti or in the poster of our workshop. They also have names, so I'm not really good with, I told you I'm not really good with names in biology, but this is the Stickus pattern, the decusate pattern, the world pattern, the alternate pattern. And you can quantify the angle that those outgrowths will do. Oh, I have a rapid growth. I actually was talking to students of school, like 8-year-olds, about Fibonacci sequence, and I did this here. So we're gonna. So Fibonacci, he had this idea that if you have young, you take a pair of juvenile rabbits, and then it grows in a season. So you still have one pair, and then they're gonna have a pair of babies. And then these babies are going to grow, right? So every season, these parents are still making new pairs of babies, and this one is growing. And so this is the sort of the thing that he thought. He thought about rabbits, and he thought, what's happening if we count the number of pairs, right? 1, 1, 2, 3, 5, 8, right? So you take the number and you sum the previous number. So that sequence, that Fibonacci sequence, has something to do with these patterns I'm talking about. Yeah? So for example, if you look at a pine cone, yeah, and you can trace out these segments of outgrowth, right, which are actually these meristems which have grown out, you can actually already appreciate that you see some swirly patterns, yeah? But there are two swirly patterns that you can see. So there's one going to the right and one going to the left, okay? So it's very nice, because this is something you can do at school. You ask the kids to go outside, get stuff, and we image it, we do some counting, and they count it, and they love counting, right? So you'll see if you count this here, 1, 2, 3, 4, 5, 6, 7, 8, you have 8. And if you count in the other direction, you should have the next number of your Fibonacci sequence, okay? You can do this with something a little bit more complicated, like a sunflower. Sunflowers, we call them planar spiral because they have this big facet. Heliolanthus, so there we go. So here, it's a little bit more complicated. So you have this direction, that direction, and you have, after a certain while, you have another type of mode. But both of them, so will be 21, 34, all three, and then 55. So again, those are the 3 sequences which follow in the Fibonacci sequence, right? And of course, you know, this is what mathematicians and people they saw and they really liked. Oh, there must be a deeper meaning, or, you know, God things in math, all those things, yeah? So, yeah, here are just some other examples of just to convince you. Artichoke, next time you eat your artichoke, have a count. But where do they really come from, yeah? So let's link this a little bit more to the biology. So yeah, so we're looking at these structures, but actually this here comes from this. So this is 2 microns. So this would be 2, 4, 6 microns. Teeny weeny, right? Smaller than a cell. But so these little bulges are already making the little seed formations on top, which will later be the fruit point. And here, they're being generated. This is where it starts. So it's at this scale that the pattern is being laid down, okay? Which is interesting, because we all have diffusible things happening. You know, we have molecular stuff at the thing. Something also very nice about Fibonacci sequence, the people always talk about the golden ratio, right? You guys know that? Yeah. So one way of looking at it is just if you take the first number and you think about the perimeter, like if you think about a square of 1, the next one, you fit it, will also be 1. But now they make it a side of 2. Their sum is 2. Yeah? And now that 2 plus 1 makes 3. Yeah? And that 3 plus 2 makes 5, and so you go. And so when you start doing that, and you draw that, and now you draw a spiral through those points, you draw this type of curve, which is called the golden curve. And it's called the golden curve because the ratio between n plus 1 Fibonacci on top of a n, as it goes higher, will give you a ratio, which is the golden ratio. And because you're generating this golden curve by intersecting the points on the series formed by the Fibonacci numbers, we call this a golden curve. And if you open up the page, the book of Darcy Thompson, here I go again, on growth and form. So this is the same guy that says that everything is surface tension. He also says that there will be something about the golden ratio which is selected for. Yeah? And he has been showing this in so many different types of systems. Now, he was obviously not the first one. So people really like these mathematical ideas. So you can go back to art. So, you know, today's a nice day. We'll have pretty pictures. So we see that we have this ratio in many paintings. People have done this consciously. Salvador Dali has done this very consciously also with left-right symmetry. But even in non-Western art, we can see it coming back. Okay, so now if you slice up fruits or vegetables, you tend to see break of symmetries of 3, 5, 8 stuff like that. Okay, not always, but it tends to be something. So we start seeing all these coincidences, right? So this is something that many people thought about. And I want to bring this the story back to Turing and to Turing's untimely death, because it's linked to this problem. So, okay, so basically, these are the Fibonacci numbers, right? What I was telling you is that if you take this ratio, you'll be able to get this number, right? This golden ratio. And you can also write a closed version here of your Fibonacci sequence with these numbers inside. This angle itself, if you calculate it out, is 137.5. So in spiral phyllotaxis, successive leaves, they grow approximately with this ratio. So when you see a meristem, 137.5, the next one comes and so forth. Now let me tell you that there's a lot of work done at the moment showing that there's quite some noise there. It doesn't always come there, right? And there's something about the average being around that. So the biology is sometimes a little bit more complicated. For example, if you really want to do a good, thorough job and go into the forest and look at your pine cones, like in Norway, the Norwegian pine fir. Okay, 95% are Fibonacci, 4% end up being the Lucas number. But now, this is actually quite similar because it just starts off a little bit wrong. So you have the L1, the first number is 1 and then 3, and then you add 3 plus 1, 4, 4 plus 3, and then you continue. So it's the same iteration, but you just have a little mistake in the beginning, right? And that beginning we can call like your axiom, and I'll tell you why. But you also have some which are not properly formed. We don't have any recognizable sequence, okay? But they still look like pine cones. So back to Lindemeyer. So Lindemeyer invented an algebra called the L systems. And L systems is another, anybody heard about them? Okay. So L systems is another modeling formalism that you can put in your tool box. And I'll show you how what it can do. Well, actually, I'll tell you what it is. It's something where you have an alphabet. It's an abstract algebra. You have an alphabet, you have an axiom, and you'll have like a production rule. Okay? And then basically, you start iterating these things, and you start writing down strings. And these strings, by applying your production rule, become bigger and bigger. Now, this is really nice to, you can, with these algebra and the correct axiom, you can get like fractals. Yeah? But you can also get something like if I say, okay, I have an alphabet which is A and B. My starting condition is going to be A. So that's my axiom. And I'm going to have a production rule, which is to say that if I have A, it becomes an AB. And if I have B, it transforms into an A. Okay? So what we do is we iterate this successively on top of the same string. So this would be my starting thing. N equals 1. I have an A and a B, right? Now, next time, my A becomes again an A and a B. And this B becomes an A. So I'm actually repeating my bunny rule, right? This one here, A, B, A, A, B, yeah? So 1, 2, 3, did I do it right? 5, yeah? Fibonacci sequence, okay? Now the cool thing is, you can also have like certain operation that this does. And so this would be plants, which are generated by this, but where one of the things you put here is like some brackets, and the brackets means make a stem or make a line, yeah? So as a kid, if you learned how to draw a tree, you say, oh, I make a trunk, now I'm going to make 2 branches. Every branch splits in 2, and every branch splits in another 2, right? This would be a very compact notation of an L system. Yeah. Okay. The nice thing is that this is algebra, so you can sort of predict and you can do a lot of maths and a lot of abstract things on top of this, okay? Here, they put some arbitrary lengths. Now, you can also say, I'm going to have rules and I'm going to use them stochastically, probabilistically. So then I can have the same rule, but if I apply it again and again, I'll get different plants. But I can still see it from the same species, because I can still see that the underlying rules and the distribution of those rules are the same, although the iterations might be different. So you can start now looking at plants and start formulating that in that sense. Or you can do something very naughty, and that's what my supervisor did. You can say that the rule is now dependent on the context it's in. Okay? So there would actually be some feedbacks, right? So you make it less abstract, less algebra, and you make it a little bit more biological. Why was that naughty at first? Actually, he really hated it when they published it, Lindemeyer, because it messed up a lot of the nice stuff you can do with it theoretically, right? Because now you have local things, and it's very difficult to deal with your strings. Yeah, because you have interactions which aren't just bindly string substitutions. But it was very interesting from a biological perspective, and it looked good. I mean, Pauline, when she made these things with Ben Hesper, you know, they were basically like drawing it out or taking pictures of a screen. It was very basic computer graphics. So Lindemeyer, after some time, got around with it and actually thought, hey, maybe this is a good idea, because now we can use the stuff that we like to describe biology. We can make pretty pictures. So there's a really nice book. This is actually seriously going through some science, seriously going through some algebra and some interesting things, but it's also showing that you can get pretty pictures. And it's called The Algorithmic Beauty of Plants. So this is also pattern formation. It's not PDEs, right? But there's a whole world out there which is about patterns, quantifying them, generating them. And this is actually when you see a movie, a Walt Disney movie, like, or even when you see The Lord of the Rings and they're like fighting in a forest, the computer graphics there to make every single tree look like a tree, but look slightly different so it doesn't make you think, oh, I'm in Uncanny Valley. They use L systems. Yeah? And they can also now use it with like, oh, there's light here, so there's going to be more cannabis growing there. You can have shadow. You can have a lot of interactions happening. Okay, so there's a lot of real science being done with L systems to really understand crop productivity and so forth. Okay? Yeah. The problem is now, if you ask your kids, that's my daughter, to go out and find, you know, these patterns, they will, but very often, it's not really going to give you, in this 5534, I think it's okay. But sometimes these dandelions, they give you something else, like 4364, but don't know what it is. Okay. Now, where these things don't work is actually very interesting. So in this case, for example, they found that the meristem itself is squashed. So it doesn't have a circular constraint to begin with. It's very elliptic. And that is actually the explanation of what is happening here and why you go off those numbers. So it is very interesting to try to understand these things. I just wanted to tell you, you can find it also in your food. So look. Now, let's go back a bit to the biology. So Reinhardt and Kuhlemeyer, so Chris Kuhlemeyer and Reinhardt, they did something really cool, which I will tell you later. They did an experiment on the meristem of tomato. They had a mutant where they take away the pins. The pins are the transporter of auxin that I told you. And those meristems are quite naked. They're not making those primordia coming out. And then they were able very carefully to put a droplet, a bead, a droplet that would stick, which was having a lot of auxin. And then this thing made primordia. So they said, haha, we apply it locally to something which doesn't have that auxin transport, and we can get an outgrowth. Okay, so who is the thing responsible for the localization of those starting points is probably auxin, right? But this was the state of art back then. They said, okay, the mathematical analysis of spiral phyllotaxis describes the arrangement and packing of the developing organs around the shoot axis, and it emphasized the regularity and precision of patterns. So you guys were telling me that patterns is about regularity and repetition. So this is a nice, well-behaved one. However, if mathematical models focus on the geometry and arrangement without considering the underlying cellular and molecular mechanisms, they remain descriptive. So you basically, they're saying, you're not explaining anything with this or whatever. You're just reformulating what you're seeing, right? Describing the rule, yeah? And so they say that, well, there are some models here which try to think about principles that could be happening. Mitchison, for example, he talks about a principle that if a meristem, if one of these primordia, which will give rise to a leaf or something, has a certain interaction radius, but this meristem is growing, then there's only some space left here where the new one can come. So he thinks about it as packing of circles which are arising. But where did he get this idea from? Okay. And so it was really, so I want to tell you a little bit of the history of this, because it's actually Turing's last and unknown work was phyllotaxis. Okay. So there's a letter he writes to Young in 1951. And so he talks about my mathematical theory of embryology. That's where he describes what a Turing instability is. Right? That you can have a well-mixed system, which its stable state will be, its equilibrium will be stable, has to be stable. And he shows what are the conditions for that stable point to be able to, due to diffusion, unstabilize. And he says that, well, you need two, he looks at it, your Jacobian, and you say, you have to have on your diagonal a positive term and a negative term, and the off, you can have either way. So the activator has to activate itself, but it could activate and inhibit the inhibitor. The inhibitor has to inhibit itself, but it could activate and inhibit the activator, right? And in fact, he has a lot of writings where he thinks about cannibals and missionaries, right? And this is the way he's thinking about it. So I leave it up to you to think who's the activator and inhibitor of cannibals and missionaries. Okay. But that's his theory of embryology, right? So it's yielding in treatment, and it will, so far as I can see, so it says like, okay, it can give some fruit. And these are the things that I think it might be able to answer. Gastrulation. Do you guys know what gastrulation is? Yeah. The most important moment you guys ever went through was gastrulation, as a gastrulation, people like say. Okay, and that's probably because gastrulation also comes with the formation of the somites, which is periodic and repetitive. So I think he was thinking about that. Polygonally symmetrically structures, starfish, flowers, leaf arrangements, so what I'm talking about here. In particular, the way the Fibonacci series comes to be involved. Like, this is really interesting. Okay, color patterns like zebras, spots, dappling. And patterns on nearly spherical structures such as some radiolera. So that's his scope. Okay. So he started tackling this problem of phyllotaxis. He worked for 3 years on this intensively, and what he wanted to do was to take reaction diffusion, those equations that you guys know, and he wanted to have it on a growing domain. Okay. And this is pretty complicated. He had to do this computationally. So some things he could already do analytically, but he knows I can't. I have to do it computationally. So I would say he was really the first real computational biologist in that sense. And, in his notes he writes, the parts are coming. So he was building his own computer sometimes. He had to build some solvers. Like, it's very, very difficult. The way the output was printed, he had to reinterpret that to get a spatial pattern. It was quite difficult. And I'll tell you why. So, as I was telling you, these plants, like the shoot, right? There's going to be a region, a dome, and here cells are being sort of made, and they're growing, they're dividing, and they're moving downwards, yeah? Can you see that? So this is growing up, it's generating things, and those cells are moving downwards. So if you would like paint something here, that dot would start moving downwards in relation to the apex. So it's a little bit like a conveyor belt. Yeah, you're always moving that. So his idea was, we run these equations around here, but they continue moving, right? And then they will dynamically still interact with one another, so then maybe new things can come out. So this is what he needed, because he knew that the Turing pattern by itself and the type of patterns it forms do not give you Fibonacci-like spirals, not at all. So he needed to mix it on a growing domain. So he has then this outline of the development of the daisy, which is part of that paper, where he's trying to formulate this. And he talks about, so this growth that I'm mentioning here, he considered this a parameter which he calls time, because this would be the time that the thing is growing and moving downwards. Okay, so then he talks about these flow matrices, which is what I'm describing here. So a convenient way of picturing these flow matrices is to imagine the change in the lattice as being due to the leaves being carried over the surface of the lattice. Sorry, I wrote this very early this morning. By fluid velocity in a linear function of position. And I have a really bad and scanned-in picture of his work. So here you see du dt, dv dt. So here you see your activator inhibitor. And here he's putting some velocity flows in. Okay? So this is what he was trying to look. Now, the people that he was friends with, after he committed suicide, they came and they tried to salvage what they could from his works, yeah? Because, you know, he was also a persona non grata at that moment from the government because he was homosexual. But they didn't really understand his notes and his jitters and everything he was getting to, right? So they never really were able to publish some things, or they tried posthumously to publish some of his ideas, but they weren't able to interpret that. But one picture that is there is this one. So he was, and people describe that in those last 2 years, he was looking at lots of printed blobs, right? And he was trying to find spirals, and he was trying to find the Fibonacci sequence back, right? And so I think he had already some really good attempts here. So this would probably be, you know, well, those are the blobs, and he was trying to look at the correlations between them if you think about these spiral lines and trying to find those things back. But he didn't really. Yeah, so there's an overview about what he was able to show or not, how far he went, by Jonathan Swinton. It's called Watching the Daisies Grow, Turing and the Fibonacci Phyllotaxis. It's really interesting. So what he was actually thinking and realizing, although they didn't have this level of resolution, is that at the very tips, what I'm telling you of these meristems, right, there's going to be actually a dome where nothing happens, and then suddenly the very small primordials come in. So P1 is the one that just came in, P2 is a little bit later, P3 is later. And depending on the sequence that they have, you can predict where the next one will be. And the rule of thumb, what you start seeing is that if P1 is here, P2 is trying to be as far away as P1 as possible. So you would think it's here. But then P3 comes into existence, and P2 has to sort of like move around. Okay? And this is the experiment I told you where you had a naked meristem because you have a PIN1 mutant, and you add a bead here, and you can get the outgrowth due to auxin. Okay, so what do we know now? We know now that, so these new primordials, they start growing at the site where the plant hormone auxin is actually is placed, not depleted. And then comes Yves Coudet, okay, which is a physicist. And he's like, I'm a physicist, but I really like these patterns. And hey, you know, there's a growing domain, and there's interaction between these primordia. We don't really know the molecular details of it. What would be a simple way that experimental physicists could go about by showing the principles of phyllotaxis? So, does someone mind lending me coming here with their laptop so I can show a YouTube movie? Because I think my laptop is going to freak out if I try to change modes. Does anybody have any questions so far? No? Okay. If you put it here and you go to YouTube, thanks so much. And then I will, because his experiments are really nice here. Coudet, phyllotaxis, a phyllotaxis of code like this. Okay, this is what I want to show. Yeah, second. It's not this one, right? Is it? Could you plug this one in, Ricardo, for me, please? Yeah? Question?
Yeah, when taking a look at the way you define the Linder-Meyer systems, they look like regular grammars in the Chomsky's hierarchy.
Yeah, exactly.
So I was just asking myself if maybe if you have a more complex grammar, for example, context-free or something like that, you get more complex patterns. Is there any study in that direction?
Definitely. So it is in the class of the Chomsky grammars. And then they do it, well, the first one I showed you was context-free. And then afterwards, it comes in all the variations. So if you look at that book, it's a really good starting point, the one from, Chemik, Przewski, who's in Canada. He actually has, he works also together with algebra people to explain how you can do it for plants. So yes, so you go to all the things. Nowadays, in fact, I will show you can even do like cellular modeling, which is on the basis, reminiscent basis of this L system structure. But it has deviated so much, because now you have local rules and things like this, that it blurs out a bit, that original beauty. But it's exactly that. Yeah, very good. It's good that you noticed. Okay. So what he has here is he has a construct where he has like a. So what you're looking is a camera which is above, looking from above. This thing here you see is just like a little tube, and out of it he lets some oil droplets fall with a certain periodicity. Right in the middle of this, a basin which is full of liquid. And these are, I think they have ferromagnetic properties in the oil. And he has this in a coil, so he has a magnetic field going up. So these particles, due to magnetic field, they want to go radially out, okay? So only one particle will go out. But because they're magnetic, they actually interact with one another. They repel one another. So you have them wanting to move out, radially out, but also having repulsion, yeah? Okay, so that's just what he did. So they repel via this magnetic dipole interaction. And here we go. So the frequency is very low. So this is what I want to say, so they're moving out. Yeah? In the beginning, it's a little bit boring. And, because this interaction is proportional over r squared, it's actually, they're not feeling each other that strongly. At a certain moment, he's going to start making that frequency of starting those of letting those droplets fall a bit quicker. And he can also change the velocity by which they move outwards. Okay, that was the first one. You get the idea. Sorry, where's it going? Oh, is your screen going dark? Oh no, maybe it's experiment going dark. Ah, there we go. Okay. Now what you start seeing is that they start. So this one moves here because it's being repelled by the others which are already moving. So they start seeking a position to move in which has the least other particles, closest particles. So they are mostly repelled by those closest to them, but still weakly repelled by others around them. And in that way, they will find a place to move radially out. They actually start curving. Okay, so now you start getting a pattern like this. So we have a switch of patterns. Suddenly, first we're all going out, straight. Now you start seeing these type of swirly patterns. Okay, while we look at this, I'm just going to say a few things. What's the analogy here with what I told you about these primordia of oxen and so, do you see that this thing was growing, right? So you are moving outwards. Yeah? Oh, there we go. So this is what he was able to mimic. He was able to mimic in a certain way that growth. And he was also saying, okay, where you have those droplets, you're going to have, that's the auxin, that's where your primordia is. They will repel each other. So the next one will go, so this is it. It will go in the space which is most in between. But this depends on how quickly they're being formed and how the movement is dragging them around. Yeah? Okay. You could also get this with a Turing pattern. In fact, you can simulate this with a Turing pattern. Because when you get those spots, and they want to repel with a certain thing, but you're moving, you suddenly have space for another one, another mode to come up, right? But then you'll have to have a position where you start fixing those patterns. You can't just randomly continue letting them go up. So if you do your timing correct, you can transform a Turing instability with some growth and some timing factors into that. Yeah? What this doesn't capture, though, is that in actual meristems, it's not at a point where it begins. It's in a certain ring. There's a certain region where those patterns are forming. Okay. So, the cool thing about these experiments of Yves Coudet, they were really simple. And he could show every single pattern we see in plants. Every single one. Okay. Just by changing very few parameters. And it was just like, I just find it very beautiful. It just takes some time for my thing to be, and he didn't stop there, right? He, so he quantifies them. He afterwards does simulations and theory to try to show how you can go from one pattern to another. Because that was another thing that confused botanists. If you look at a small scale, you have one pattern. At a certain moment, the pattern changes. So he could show how you can bifurcate from one of those modes to another. So all of those transitions he could show. He could show how suddenly you can go to Fibonacci numbers and why that's more stable, and how you could also end up in Lucas numbers. Everything could be explained. Yeah? Yeah? Yeah, you get different type of patterns. And that's what we're doing nowadays, biologically, because there is something which tunes the timing, and that's a hormone called cytokinin. Yeah, so we can change that. Very good. Yeah, okay. And, and he can look here, golden angle and so forth. So I highly recommend you guys to have a look, if you're interested in this, into this paper. This is where he's actually really working out the theory. And here he also then goes further to say, okay, let's take our experiment, which was a very idealized situation, and let's think about it indeed, that we have a meristem of a plant with a certain shape. We have this region here, where we know that in this interface, your meristems can start. And we have these different periodicities, and we have these different growth of velocities, and we might also have different repulsions between the droplets, because those are the ingredients you need. And again, he can then show all of the different type of patterns we see in nature. So it's very nice because there are lots of mutants which do lots of crazy things, but now we narrow it down to think about growth, periodicity of formation, and repulsion. Okay? So how does repulsion really work, though? So then some people started thinking, well, it's auxin transport, right? So if you start having a little bit more auxin somewhere, and a cell has a little bit more auxin than its neighbors, okay? This one has a little bit more than here and here. And so I'm going to just draw it like this. The rule is the following. You have cell A and you have cell B. Yeah? A has a little bit more auxin than B. So B is going to start, B knows that. B takes the difference of its auxin concentration with that of the neighbor. And it's going to start directing its pins to A. Yeah, so what's going to happen? So A is going to get even more, right? And then the other neighbor will start doing that. And the other neighbor will start doing that. Yeah? So then computational model said, okay, now we're going to put the biology in. The biology in is that pins start orientating themselves to the place where this primordial will form, and it has a knockout effect that is quite long range, because the next neighbor will also point to here, because this one, right? And it's going to go up. Yeah? So at a certain moment, they have certain cutoff values to keep that range intact, and then a new primordial can go somewhere else. So they can sort of mimic that repulsion with this thing. And these would be the PNS papers coming out from there. So one is those like spherical spheres. This one is a L system type of model. And here you have this. So these bubbles that are being generated, they're moving downwards. These are cells. They measure each other's concentration, and they give oxygen to the cell which has more concentration than themselves. And so in that way, you will have regions which will have high concentration and regions where it's depleted. And in these regions where things are depleted, a new maximum can come up. Okay? And they also can change the growth rates and also the interaction of these pins, and they can get different forms coming up. So the problem here is, has this taught us more than those other models? I actually like these models, yeah, because you're putting, but you guys shouldn't be always dazzled if people put molecular things in to say, okay, now it's more realistic. Because it's still having the same components as the magnetic field, which is obviously not a plant, yeah. But it is sort of telling you that now it's all done because a cell measures the other concentration. But how do you do that? How do you, as a cell, know that the other cell has 0.0001 more than you have of oxygen, right? How do you measure difference of concentration when you can't even touch that cell? You know, where it's in the cytoplasm, yeah?
And that's where the new growth happens. Like, wouldn't that just get damped? Like, where's like, are they just constantly producing? And
yeah, because it's about the relative difference between the neighbors. So because at that region you don't have any more like that they're moving it to the maximum. So you have a region where everything is sort of flat. The new maximum will be away from moving it to the neighbors, right? No, it's not a maximum yet. It's just going to be a small variation which is going to be picked up stochastically.
Sure, okay.
It's very numerical. I agree it's confusing because,
Are you done?
No, go ahead.
What is the time scale at which this happens?
What is the timescale that this happens? That's a good question. Usually in real plants, I think every meristem gets, at that moment, gets laid down every hour and a half, 2 hours, and then grows, yeah? And sometimes a little bit.
I meant the time scale of basically this information that they're processing about their neighbor's concentration.
That happens like instantaneously in the simulation. Yeah. So the more pins you localize, the less oxygen you will have, right? So if you're close to a maximum, even if you're in the neighborhood, you will have very low amounts. After that effect is dampened, which will be away from them, that one will be able to have a little bit more, because it's not expressing its pins. It's a very artificial threshold. So it'll have a little bit more, and then those neighbors will actually point to him, and then it comes up. So this is the problem. We don't know how oxygen and neighbors can be sensed and how you can take a minute difference to start this up. And what we start realizing is that why a cell polarizes in a certain direction is really the key process of understanding these connections, right? This tissue polarity which is ensuing. So nowadays, I would say we're still quite unsatisfied because we still don't have a molecular explanation of the autoemergence of how we do know the pins are polarizing. We know that's happening, but we don't know how it's felt or what's starting that process up. Now, some people are saying that there is actually some other genes which are rising in those points, which might be sort of reaction diffusion and Turing pattern-like, and that the pins might be picking that up. Or maybe it's an interplay, yeah? So, for example, this is a more recent study, which is saying that one of these transcription factors which regulates cell polarity factors, right? It is actually very key to be able to have that cell polarity oxygen feedback, to localize a neighbor to a cell which has a lot of oxygen. Again, it's still unclear how oxygen would be felt in a neighboring cell, right? But it could be indirectly, because if you have more oxygen, you know, the state of the neighbor would change, and that could indirectly give some type of signal for the information to put up. I'm really sorry I can't give you a better explanation, because there is none. It is easy to make models now, which can generate it, but it is very difficult to give a molecular explanation. And what's also very difficult is sometimes these models, because they're very rule-based, they have thresholds and so on, it's also very difficult to precisely understand what they're doing. Whereas I think Eve's model is still very pure, and we really understand at least the abstract components. So Eve himself was still working with many people to try to understand these things better. So this is Eve at an EMBO course that I organized. And this is him actually giving a talk in the bottom of a boat in East Anglia, discussing about quantum dots, so something totally different. So yeah, so this is what I wanted to tell you guys, something a little bit different, just to zoom out and to say that there are other ways also of analyzing patterns, of modeling them, and they beg questions, yeah? And phyllotaxis is a big one. So, now it is almost 10, so I have 45 minutes. So I'm going to quickly have a little pause here, and I will open, was this one open? Yeah. Okay. Oh, just a second. I need to. I'm going to change the display. Oh no. Wasn't this? Was it? Okay. Ah, fuck. Sorry. This is being recorded. Sorry, guys. Acroread. So that's the first part of the talk. Of the lecture. Okay, and now, sorry, there we go. There we go. Okay, so this was the story that I told you the other day when we were talking about morphogens and how we can look at transport-mediated reflex loops and roots, and why it's so important to have that lateral flux. And the thing can grow. Okay. Yeah, and so this is where we stopped. So to link this back to that story, again, we're going to have the pins, we're going to have oxen transport. Now, one thing that I always tell my students, we will never build a model where we assume something that we know cells can't do. So a cell cannot read a concentration in a neighboring cell. Yeah? I think there might be other ways that you could get something which, on a higher level, might simulate that type of behavior. But if you really want to say that I'm trying to do something which fits my experiments, you should actually try not to cheat. It's very easy to do that as a programmer, to have the information of the states of your neighbors. But this is exactly what you don't want to do when you think about developmental biology, because it's very local, right? Somehow that information comes through. And that's the question which I think is really important to address. And if you really want to cheat, then say, well, there's a long-range interaction. But then do it clearly and say you're not doing it with the pins. Yeah, this is my personal way to go with it, because it does change a lot of stuff. Okay, so when we talked about the reflex loop, yeah, the nice thing is, although it was very complicated, because we have many cell types and we have many polarities, but we could see that this is what's happening with the flow, right? There's a huge flux is right at the tip, and everywhere this is leakage back. And that is the reason you have an exponential gradient, not because of how much you're pumping down and up. You start seeing this image in many other systems. And so this is why I think it's also very important, after you do some modeling, to try to abstract it. So this reflex-driven maximum, like it becomes like a concept, and you can start, through what I would say is isomorphisms, you can start seeing them in other systems. So this paper, for example, it shows, the question is, how does prebiotic evolution start? Because you need quite a lot of nucleotides together for those reactions actually to start up. How would that have happened, right, in this world where everything is like diffusing around? And one idea is that at the bottom of the seabeds, you'd have these small little ditches, yeah, very small. And because it's volcanically active, you would have convection because you have temperature gradients around them. So those would be typical structures they would have seen in those times. And you would have thermodiffusion due to those. So you have convection, so water is coming up, the flux is coming down and up, and then there's some lateral flow. And they say, okay, if you combine something coming down and up, or with some laterality, you're going to get the accumulation at the tip. Yeah? And the longer those tubes are, right, the more that you're allowed to have that laterality, the higher those concentrations will be. So those are the things that, you know, they're just very nice principles. Also, your kidney has to, in the end of the day, it has to take away toxins, right? Has to do filtering. So it has these structures which go and come back. But at the tips, you have very high concentrations of your toxins, but you clear. And the questions people had a lot of time, how is that getting accumulated? Because they didn't see the mechanism. Because the only thing that they had there is leakage between those currents. And what they realize is if the leakage isn't there, if you don't have the connectivity between those currents, you wouldn't have that accumulation, right? So those are insights that you start building up, right? Of course, the reflex loop, because it is very quickly making a gradient, right? And as the route elongates, because transport system is quite fast, so the permeability is around 20 microns per second. So this is the permeability rate of those transporters as measured when we express them in an oocyte of a frog, and we look at transport dynamics, so we can actually biochemically measure these things. It means also, and because the fluxes are so grand, so great, that as the route grows, you don't have that dilution effect. It's really just moving with it, right? Because the polarity is kept. So it's moving. And what you start noticing now, so this is how the model works, is that when you have high concentrations, you sort of divide. As a cell, when you start encountering these lower concentrations, you elongate, and at a certain moment, you differentiate. This is the way we thought about it in the past, right? So from the perspective, if we now change our reference system, so the tip is always at the same point, and we're looking at the flow of the material in the root, we see that this cell is moving across the gradient. Okay, so our meristem zone slowly grows over time, which is something we see in the first few days. Okay, so that's sort of concluding that. Now, what I want to do with you is show how, although we're happy, you know, we can do a lot of things with our experiments, we understand the system, we don't really understand the question, why does it do it like that? Okay, and this does sound like a bit esoteric question, but it is a very important question sometimes to ask in biology. And I'd like to quote Eric Lander here, because he works a lot on morphogen gradients as well. But he does also something very interesting. So he says, well, to answer this qualitative question, so why does an organism do it that way? You only really can succeed, at least in many cases, if you take a quantitative approach, right? So in this case, and this is very sort of easy mathematics, because the only thing we're actually going to consider is, okay, so these are the simulations of the reflex loop, where we know what the diffusion of oxygen is, 800 microns squared per second, and this permeability rate of 20 microns per second. So these would be the typical gradients, and this is the characteristic length, and we have a certain slope, right? And we know that this is very good to reproduce these type of patterns we see in the root. So now you can back engineer it. You can say, okay, because some people were saying that the way you can get a gradient in roots is by producing it at a source and degrading it somewhere else. So what if you put a source here of auxin? Yeah. This is the only source. We have degradation everywhere. As we know, auxin is degrading. And we use the same parameters, right? Actually, it's 600 microns squared in this simulation. 600 microns squared per second, and 10 to the minus 6 is the breakdown, okay? So what you see is actually because you have such a high diffusion and such a low breakdown, you're going to get quite a linear gradient, right? So this you can do analytically, obviously. So we have, I mean, analytics, just a e to the minus square root of delta over d, x. So we see that the characteristic length for these parameters, it would move from 100 microns to 2.4 centimeters. So there is no patterning possible, right? There are no differences in concentration. Okay. But you can say, but let's say I do have that system. What parameters would I need if I were to want to adjust it to that slope? Yeah? Okay. So to be able to adjust it to that slope, we would, in this case, I'm saying that the degradation is now very big, okay? So the black line here, this blue line here, would be the default parameters, and in red here we would have just a source decay system, right, which gives you exactly this characteristic length because we calculate it out, and we would have to have a half-life of 11 seconds. So it's broken down very quickly. We try to put a big production, right? So indeed, this is the just in the simulation to see how it looks like with all the tissue types in it, we have a very nice sort of type of gradient. The problem here is that the auxin pulse, so if you have a little auxin pulse, it won't be able to travel very far, 140 microns. And those cells are about 300, 500 microns. So things communicate via auxin, but auxin pulses wouldn't be able to be transported because they would be broken down before they could reach anything. So we start seeing here that we have a problem. If you want to adjust the spatial length, you have a problem, a trade-off here with the temporal length. Yeah? Okay, this is just experiments where we make those cells suddenly produce auxin, and we can see, you know, how the expression of auxin genes turns out on. So we know that pulses, local pulses of auxin, are carried on quite far. Yeah? So it's in conflict with what we know the system is doing. Okay. Another experiment that would show that this would be very difficult is if you would stop the influx and you ablate the cell. So we don't let any auxin come in from the chute, yeah? And we would have to ablate this. And we do that with a laser. So we point the laser at the quiescent center, and we zap the quiescent center, so it just becomes a bit of debris here. And what you see is that following it, there will be accumulation of auxin, and there will be a repatterning of that stem cell. First, auxin comes up at high levels, and then that triggers the redifferentiation of differentiated cells back to the stem cell. Okay? And that happens, yes? Yeah. So, yeah, so in the model that we have, our simulation, we said that what's keeping the maximum there is mainly due to the reflex system, right? And auxin is provided from the shoot to the root, and there it's just, you know, flowing around. When we severed the root away from the shoot, so we don't allow that influx, although in the model we didn't have any production, we see that the quiescent center is still quite happy at maximum because all the auxin is reshuffled toward that center, and although on a long time scale it's degrading, right, it will still be high patterning-wise at that location due to the pattern of fluxes. So in our model, no, it's not produced in the quiescent center. And what I'm showing here is that although people see a maximum, I think that's where it's being produced, we are showing that if that were the place, if it's a maximum because of production, none of the timescales would make sense. Because when we ablate the cell, if it were the producer, it wouldn't be able to reproduce auxin somewhere else on such fast timescales. So the only way to predict this, especially if you have severed it, is the fact that we have reflux happening. So this experiment was a way to show that the auxin that's accumulating at that intersection, so this is what the simulation does. We ablate it also in the simulation, so we don't now have those cells, so nothing is able to pass. And what we see is very quickly, we have the reestablishment of the maximum on top, because the reflux loop is intact. You might have killed those cells, but you're going to have a new accumulation. And that new accumulation will trigger the new cells. So having a reflux loop, instead of saying the maximum is the one who produces the cell, it's sort of healing itself. It allows you to reestablish new foci point. In fact, it's even worse. If I cut the root here, a new maximum will appear here, right, because of the influx. However, if I do cut a piece of root, and it's just very small, so I don't have much auxin in it at all, there's not going to be enough to accumulate, so I don't get a maximum. It has to be a big enough segment of root. And the cool thing is if you take a small segment of root and you put it in a bath of auxin, you put it in a, right, in a little thing with auxin, then the auxin will be able to enter, and then you'll be able to accumulate it at the tip. So what we're showing here is that auxin accumulates not because it's produced at a location. That's not how the patterning is done. It's done because you have a structured pin polarity in your tissue which allows it to accumulate at a certain position. So it's a different way of thinking. Yeah. Well, in this model, we have diffusion because we have the spatial scale of the cell, right? So in the cell, you'll have a flux due to diffusion, right? But it leaves the cell due to permeability, pump-mediated flux. So here the J, the flux, is the permeability of these transporters times the concentration inside, yeah? Whereas here, your J is just, right, the diffusion. Yeah, it's also diffusing all along your cell walls, which is an apoplastic space between cells. So auxin is able to leak around still by diffusion in this grid, this mesh of cell wall, right? So it doesn't go directly from one cell to another. It first goes into the cell wall, and then the next cell will take it up and so forth. Depends. So for auxin transport, it's really the dominant mechanism is the permeability. But leakage is very good because it allows to connect flows. So we know it's quite interesting. It has a different scale. And we have some studies now that we show that nutrient transport, although we know they're transporters, the cell wall plays a really important role to be able to understand the flows that we get. So we actually can't always simplify the simulations and just say, oh, let's just have the cells, and let's forget about the cell wall. Sometimes that small space in between really changes the type of dynamics, even when you have a very dominant flow. Okay. So yeah, so obviously, so the morphogens root mean square displacement, right? In a source, decay, reaction, just diffusion system, right, is determined here by the diffusion rate and the degradation rate of your substance, right? You can't get away of this hard constraint. But you can, though, if you have a pattern polarity. Yeah? Okay, so, okay, here we did other things. We localize sources somewhere else, right? So we have, in this case here, auxin is being produced in other cells, and we see it accumulating here because you have a transport system. And in this case here, we have auxin produced everywhere in the root, and we see it still accumulating at the quiescent center. So where you produce auxin, you don't see it anymore because it's quickly being taken away, and it's accumulating at the quiescent center. Okay, so now you can say, okay, but you know, Veronica, we know that plants have transporters. Yeah, pins are around the whole time. Obviously, it's not going to be just degradation and diffusion. So why not just have a linear transport? Why wouldn't you just have said, this is a donor cell, like your shoot or someone who's producing auxin, and you're pumping it forward to the end, and you accumulate it here. And in fact, very primitive structures like mosses, they do it like this, okay? They have like a polarity like that. So let's say that the root was just like a vasculature, everything moving downwards, right? Why have that flow up and back and cycling and all that sort of complication? So if we have a system like this, we can also, so basically you'll have a concentration here, you have your pin permeability downwards, we have leakage terms, yeah? In this model, by the way, just to do the maths a little bit more easily, I did forget the cell wall. But we have also other versions with the cell wall, just a little bit more complicated, but it gives a very similar end state. Okay, so then we'll have a flux down, we'll have a flux up, right? And then we can equalize them to see how the pattern will look at equilibrium, right? So if we would wait long enough, this would be the equilibrium pattern. And so what we see here, it's going to be exponential. It's going to accumulate here at the last cell, but it depends on the number of cells you have in your row, okay? And this parameter, lambda here, depends on this ratio of your permeability rate, so what is your pump doing, and what is your leakage term doing, your back leakage, yeah? Okay, so if we put the permeability rate, which has been measured in the laboratory of your pins, and in this case, we only have this sort of vasculature, everything is pointing here to the tip, what you see is you get a gradient, but now the problem is the gradient is too sharp. It's way too sharp, okay? So we have a gradient over the first 4 or 5 cells, and then after that, everything is, you see here, it's linear. Remember that these sawtooth that you're seeing are the patterns within the cell because you have diffusion in the cell. You have a flux and you have diffusion in the cell, so you have those concentration differences. And this is very bad because we know that our meristem should be around, 300 microns, and we get here the elongation zone, so we need to have a bigger characteristic length. So then you can say, okay, we have this mechanism, so let's back engineer it. Let's decide what are the parameters we need for permeability. We don't have to be that good at pumping things forward. We can just pump it a little bit, okay? So let's find out what the p-value is, right, for the characteristic length to be exactly what we see in our wild type, in our full model. And so what we see is that, so if the pin would be like 20 microns per second, our leakage term would be 17. Okay. And then you get this red graph here. But basically now what we're saying is like you take a step forward, like 2 steps forward, and then you take a step back. 2 steps forward, 1 back, because you're leaking a lot backwards as well, right? So they're very similar, your pumping and your leakage term. Okay, so yes, you do get that characteristic length, because I mean, we engineered it this way, but what we start seeing here is that time scales become very, very slow. So if you want to move something around information-wise, you're moving forward, but you're moving back on the same time scale, okay? So we lose again the time scale ability of a pulse of oxygen to travel, which we have experimentally verified before. So we start realizing that the reflex loop is a really nice system to quickly reestablish a maximum, to keep a characteristic length, to be a little bit independent of your diffusion rates, and to still be able to have a fast communication between cells. Okay, so you're able to control that. This is just to show you that this reflex system, it's quite robust to changes in the permeability rates. Also, if you would say that your pump saturates its pumping activity, its permeability rate, depending on the concentration, because it might saturate, we see that we would get differences maybe a little bit here in the maximum concentrations, but our profiles would still have the same characteristic length, same slope, I mean, because the slope is totally determined by the ratio of upwards and lateral pins. Okay, so how are we with time? We have 50 minutes. Okay, so I hope I could convince you that we start, at least, we working with plants, started to appreciate better how the plant is structured when we started to think, why didn't it do it in a different way? Okay, after all that work, an experimentalist came to me, Sabrina Sabatini, and she showed me this result, that yes, the meristem is growing, as our model predicted, but at a certain day, and this depends on the lab you're at, it totally halts, okay? And this was against our expectations because we would have imagined that this slow time scale would proceed throughout, okay? And I'm just wondering how to tell the story because I don't want to overload you guys with too much biology. But what she sees here is that there's another hormone called cytokinin, which is not transported polarly, so it's more like a system parameter, but it can be put up and down, depending on environmental signals. And it's underneath that signaling cascade of that hormone, cytokinin, there are these transcription factors, cytokinin signaling genes, which are located usually at this transition zone. And the transition zone is where cells are small and where they will start becoming bigger. So this is where the meristem stops and the elongation zone begins. So we call that a transition zone now. And this is the cool thing, that if you mutate those genes, which are listening to cytokinin, suddenly that meristem becomes bigger. Okay? So seemingly there's cytokinin there, and what is cytokinin doing? Due to those signaling, those transcription factors, it is halting. So auxin by itself would be expanding and would want to go up, and cytokinin says, no, you stay here. I want the transition zone to stop, okay? And indeed, by the way, also if we overexpress these things, as you can imagine, the transition zone becomes even closer to the tip. So we have something which is establishing where that demarcation line will be. So to understand what was happening here, so we actually made a more sophisticated, still a geometric route, right? We, again, looked very carefully at all the pin expressions and the importers, but we realized something else that our initial model hadn't shown, hadn't taken into account, that the cells at the tip, they have sometimes stronger expression of these pins, and here they have somewhat lesser expression of those pins. So this would be the classical model I told you guys about, which is just reflux. This is, again, the reflux model, so you still have the quiescent center. Everything here at the tip looks okay. But what you see is because the pins here are a little bit more depolarized, are not as strongly expressed due to those cytokinin transcription factors, the concentrations go back up. Okay? So now what we have, and this was nice because this was where our model was conflicting with the results. So if this is the tip, right, this is the quiescent center location. I look at my profile, I had a maximum. I used to have just exponential gradient. Now we have this, and suddenly we go back. And right at that region, we started noticing, hey, there's a little blip. It's a little bit of blue. It looks a bit like the lowest amount, like a minimum. Yeah? And they started looking here. So this is the inverse reporter of, actually, in this case, it would be of auxin concentration. And they see that here, at this location here, if you start looking, you always have the lowest expression. So low amounts of auxin. So we started mathematically looking at this in 1D. So we have models here where we have everything now. We have the cell wall, we have diffusion. We also, in this case, include symplastic transport, so that there are bridges between cells, right? And the partial differential solutions that you're going to get here, they're always composed out of these two envelopes. So there's going to be, here's the tip, yeah? So there's going to be this exponential part, and there's going to be this other part here, right? So you have an elbow, so it rises up. And the positioning of this elbow is very much determined with the degradation rate of your substance that you're transporting. So we talked to the experimentalists and said, okay, degradation could really locate this here more closely to the tip or further away. Why would that be important? Because maybe our hypothesis was now a minimum in our gradient could tell cells when to stop dividing and when to start differentiating. Okay, I'm not going to go into this, but what they see downstream of cytokinin, so they did chip-on-chip in collaboration with a group in America, they could see that degradation genes were starting to be expressed. So you are degrading auxin, and you're degrading auxin. When we put the degradation up a little bit, what we see is that that pattern now has a more pronounced minimum. So this is where the minimum is located. Obviously, what you need to do with a model is, okay, so we have no cytokinin. This is our control. With cytokinin, we have a very small faint dip, right? And with the cytokinin control and transport and degradation, we have a full-fledged minimum. Okay, so now our hypothesis is, well, you're at high amounts as a cell, so you're happily dividing. At a certain moment, you go from high, low to high. And this happens over time because the root is moving, right? So you're moving through this. So we don't know molecularly how this would be possible, but if you, in a certain time window, have an increase, you come from a level, you have a decrease and an increase again, that would start promoting you to undergo a differentiation process and elongate. So the way to test this hypothesis is then by experiments. So here, this is a root which is insensitive to cytokinin. Yeah? So wild type would have a meristem like this. In this experiment where you're insensitive, your transition zone has moved. So what do we do in the simulation? In the simulation, we just don't allow the degradation to happen. We also don't allow the depolarization to happen in the same way, so we lose the minimum. But under those parameter conditions, if we were to increase the extension of where we have the meristem zone, our minimum reappears. Okay. Although this is not a growing root, we can change the layout with the same parameters and show that now we have the appearance of very low amounts. So in this case, a cell would go from high, low, high again. Conversely, if we now overexpress cytokinin signaling in these roots, we get a dip of your meristem. Here, if we have a lot of breakdown, we're going to have a big zone of blackout. So it's not a minimum anymore. You're going to low values, low, 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 low. So that wouldn't be the response. So we would imagine that a cell here would already start transiting. So here you see, with a shrinkage of that meristem zone, we again regain a characteristic, that second curvature that we're looking for. Okay? And overnight experiments, this is a wild type. So this expression here shows you where auxin is high. So this is, so in this case, if it's expressed in a high way, it's showing you low amounts of auxin. So this is a low amount of auxin, a minimum. And if you now express it with cytokinin, which is you're going to have degradation happening, your meristem is going to get shorter, you see that not only did your meristem get shorter, but before that, your minimum moved. So we can experimentally also see that move of the minimum. Okay, we do a lot of perturbations with mutants and so forth. But what's cool now in this idea of morphogen space and stuff is that, okay, before we just had this auxin profile, we realized that cytokinin is interacting with auxin gradients by changing the pin polarity and by changing auxin degradation. And it morphs it in such a way that while the tip is still okay, it allows this appearance of a minimum in this gradient. And we think that this is developmentally instructive because any alterations we do to shift that minimum forward or backward, we get a correspondence with what's happening with the meristem itself. So this is my last slide. So although what the gradient wants to do is to expand, yeah? Right? We have a mechanism which is going to halt this and retract it to keep it small. Yeah? And the question is, why would you do this? Why would you be halting your meristem? Why would you be keeping it small? And the reason you keep it small is because when roots are, they're growing with a steady velocity, they're very happy. If suddenly they have to grow quicker because there's a nutrient availability, they can release the brake, very quickly expand the meristem, and grow fast. So they actually prefer, and this is something we see repeatedly in these control systems, you are able to do something much faster, but you're usually like a Formula 1 driver. You're going really fast, but you put your foot on the brakes when you do the turn. So afterwards, when you take it off, right, you are accelerating again. You don't have to re-accelerate. You just release the inhibition. Okay? So with that, I just want to finish the lecture about plants, about from phyllotaxis and back to how you can take a model and explore also why that model works, and start also, and very important, always look for cases where your model prediction is refuted, where there's something which it doesn't agree with, because then something really new might come in, which is conceptually interesting. So we find the idea that a minimum could be developmentally important for patterns in cell response quite interesting. So yeah, so with that, I think you guys have still half an hour and 5 minutes for coffee before Pierre will start. So thanks a lot.
Thank you, Veronica.
But are there questions? Because we have time.
Is there any question?
Good.
No? Well,